Hi everyone, welcome to
amazing channel of Brilliant Katha.
When you're rubbing one body with another body, electrons are transferred from one object to what? Another object. Okay, this process, this type of electricity is been given a name.
halogen connected carbon sp3, then it's connected to benzene. See all of you, halogen connected. What is this carbon hybridization? Full single bond sp3, see then a benzene ring.
Denominator poles shouldn't be included in the denominator poles. That means the denominator, these -1 and -5 should never be included in the solution.
Homocircle caudal fin. It's giving two equal lobes. But in case of cartilage fishes, look at here, one big lobe is there, one small lobe is there. You cannot cut into two equal half this fin.
Hi all, today we will be starting with the important question discussions or the important topic discussions from Principles of Inheritance chapter. So we will go through some MCQ as well as we will consider it as a revision also. So let's start with Principles of Inheritance, one of the important chapter in biology, grade 12. First question, the genotype of a plant showing the dominant phenotype, it can be determined by, suppose if you are getting a plant with a dominant character like tall, and if the genotype of that is asked, how you can tell? By looking at a plant we cannot understand. So what is done is, this dominant character will be crossed with a recessive parent. What is the recessive character? Dwarf. We know already the genotype of dwarf, that is always homozygous. So homozygous dwarf, when it is crossed with this dominant character, if you are getting all tall, then we can tell the genotype of this is homozygous. But instead, if you are getting 50% tall and 50% dwarf, then we can conclude the plant that you have taken is heterozygous. So like this, a dominant character's genotype can be interpreted using the cross like this. This is called as test cross. So answer to this question is test cross. So genotype of a plant showing dominant phenotype can be determined by, because if it is dominant, it can have either homozygous condition or heterozygous condition. If it is homozygous condition, you will get only that particular character when you cross it with the recessive parent. But if you are getting 50% dominant and 50% recessive, understand the plant picked by you is heterozygous. This cross is called as test cross. This cross is actually called as test cross, and the ratio of test cross is 1:1. Hope you got it. Now the next question, which of the following is incorrect for garden pea? So here so many characters are given and its phenotype is also given. You might have learned the 7 characters of the garden pea that was actually experimented by Gregor Johann Mendel. So in that first character that is written here, seed color. Seed color, if you are taking, yellow is actually dominant in the seed color. So in the seed color, dominant is yellow and the recessive is green. So dominant is yellow and the recessive is green. So always we will be using capital letter for denoting the dominant one. So we can write the yellow with capital Y. So it can have two genotype, either homozygous or it can be heterozygous. And the green you can see, but always it will be homozygous. So seed color, the option that is given, the genotype, that is correct. Then second one that is given is port color. Port color, if you are taking the homozygous port color, if you are taking, you can see dominant is green and recessive is yellow. So it can be capital G, let's use for writing the port color. So we can use capital G, or else we can use capital G, small g. For yellow we can use small g also. So like this we can use the port color, the genotype also. Hope you understood. So second option is also correct. For green you can use either homozygous condition or you can use heterozygous condition. Now coming to flower position. Flower position you can see it's given terminal. Actually terminal is homozygous only because terminal is a recessive character. So flower position, when you are taking, the dominant character will be axial. Flower position, dominant will be axial. Dominant will be axial and the recessive will be terminal. So terminal, if you are taking, you can see it will be only homozygous condition. Heterozygous condition will not be present. Hope you understood. So this third option is actually incorrect. So seed color, if you take yellow and green will be coming. Yellow is dominant, green is recessive, and yellow can be written either in homozygous or heterozygous. Port color, if you take, green is dominant and yellow is recessive. So green can be written in homozygous condition or heterozygous condition, and yellow should be homozygous. Then flower color, it is axial and terminal. And axial, since it is dominant, axial can be written as either homozygous or heterozygous, but terminal you can see it is always homozygous. Then port shape, constricted only homozygous because constricted is recessive character. Inflated or full is the dominant character. So 4 characters you already got from this question. So remaining 3 also let me tell. Stem height, tall and dwarf. Tall is dominant, dwarf is recessive. Then the flower color, violet and white. Violet is dominant, white is recessive. Then seed shape, round and wrinkled. Round is dominant, wrinkled is recessive. So that's all about the 7 characters in the pea plants based on which the experiments were conducted by Gregor Johann Mendel. So based on this only, Mendel has conducted the experiment in order to learn about inheritance mechanism, how the characters are transmitted from one generation to the next generation. And he was very successful in finding how these characters are getting transmitted. And in these 7 characters, when he was conducting the monohybrid experiment, he was getting 3:1 phenotypic ratio and 1:2:1 genotypic ratio. And from his monohybrid experiment, he actually said 2 laws of inheritance. One is law of dominance and the other is law of segregation. In that, law of dominance had many exceptions, but law of segregation is universal. Next question, the period for Mendel's hybridization experiment. Can you please comment what is the answer for this question? You may be knowing about Gregor Johann Mendel from Austria. He is considered as the father of genetics. He was actually bringing a lot of research in the inheritance mechanism, how the characters are getting inherited from one generation to the next generation. He is from Austria, born in Austria in 1822, and he was influenced by many research works that was actually proving about inheritance mechanism, how the characters are inherited from one generation to the next. But no one was giving a good explanation, so he conducted a research in pea plants to learn about the character transmission from one generation to the next generation. And after having a 7 years of research on this inheritance mechanism, he was able to give a good explanation. So when was his experiments conducted? Okay, one answer was given. Yeah, it is correct answer. The answer is 2. 1856 to 1863. This was the experimental duration of Gregor Johann Mendel. So he was putting research on Pisum sativum or pea plants from 1856 to 1863. After 7 years of research, after completing a research, he was actually doing his presentation. On completing a research, you can actually put your research under universities and all, and you can go for a presentation. He was also doing the same in 1865. So it was done in Natural History Society of Brunn, where he was doing his presentation. But unfortunately, his work got rejected. Even if your work is rejected, you have the complete rights to publish your work. So this person, even though his work was getting rejected, he published his work in 1865 in German language in the magazine of Natural History Society or Science Scholars Society that is located in Brunn in Austria. And in 1884, what happened is, unfortunately, he passed away. But why his work was rejected also, you should be knowing. There is a reason for his work's rejection. The main reason, his explanation about transmission of characters from one generation to the next generation was very, very far ahead of the time in which he was living. So he was telling at that time, characters are controlled by discrete units called factors or genes that we are calling now. So he was telling it a long back. At that time, the nucleus, the DNA or the gene in that was not discovered. At that time only, this person, after doing research on pea plants, was telling characters are controlled by 2 discrete units that is called as factors. Anyway, his work was not accepted, it got rejected, and it took around 33 years for the rediscovery of his work. And in 1900 only, his work was getting rediscovered. So if anyone is asking why his work remained unnoticed for 33 years, the answer is lack of communication. His work was not published in English, it was actually published in German language, and he actually published this work in a magazine that was less circulated in Austria. This is the reason actually his work remained unnoticed for 33 years. After 33 years, the 3 scientists actually came across his work. They were knowing German language. One is Schermak, and the other is Hugo de Vries, and the other is Karl Correns. These 3 scientists from different parts of the world, that is Hugo de Vries from Holland, Karl Correns from Germany, and Schermak from Austria. These 3 people were researching on inheritance mechanism on different plants, and they all came to the same conclusion as that of Gregor Johann Mendel. That is why he was considered as the father of genetics when the new branch of science was evolving in 1906. So hope you got a brief history of Gregor Johann Mendel and all the timeline that is given in NCERT. 1865 was his presentation and publishing, and in 1990 was the rediscovery of Mendel's work. Now choose the correct option based on the following statement. So here we are going to discuss about his work and his presentations only. So he was conducting first research on the monohybrid experiment. That means at a time he was learning inheritance of one character. I said 7 characters in the beginning. In that 7 characters, he was actually learning only one character at a time in the monohybrid experiment. So there was an experimental procedure. You know that the first step in his experiment was selection of pure breeding parents. So he was taking a pure breeding, which parent? A pure breeding, for example, stem height. He was taking a pure breeding tall and a pure breeding dwarf. This was the phenotype of the parent, a pure breeding tall parent as well as a pure breeding dwarf was taken. Then he was not knowing the genotype. So here in this pure breeding tall and dwarf, he was conducting a cross. He considered one plant as the female and one plant as the male parent. So in order to get accurate result, he was actually focusing on certain things like the dwarf plant that he was taking. If he was considering it as the female parent, in the bud stage itself, he was removing the pollens from that. So why he was removing the pollens in the bud stage? This process is actually called as emasculation. So you should understand, the plant that he was considering it as female, he removed the stamen that is in the bud stage. So removal of stamen in the bud stage is actually called emasculation. There is a purpose of doing this emasculation in the dwarf plant. The reason, in order to avoid self-pollination. He wanted to get accurate result of these two parents. That is the reason he was removing the stamen in the bud stage. Second, what he was doing is he was putting a polythene bag around this flower. That is to avoid cross-pollination from unwanted plants or some other plants till this flower is maturing or till the pistil is maturing. He put a polythene bag around that. The process is called as bagging. So hope you understood these two things. He was performing emasculation followed by bagging. So emasculation will avoid self-pollination, whereas this bagging will be avoiding cross-pollination from unwanted plants. Now, when the flower is matured, he removed this polythene bag and he will be taking the pollens from the male parent and dusting it on the stigma of female. This is called dusting. And once the dusting is completed, again the polythene bag is put over. So these four processes only you might have learned in sexual reproduction in flowering plants, artificial hybridization. The same step was done by Gregor Johann Mendel. So he was taking a dominant plant as well as a recessive plant. That means a tall and dwarf or a round and a wrinkled. Like the two plants were taken. One which is considered as female, in that only emasculation is done, bagging is done. So why emasculation? If you ask, the answer is to avoid self-pollination. Why bagging? In order to avoid cross-pollination from some other plants. And once it is done, when the flower is maturing, pollens are taken from the male parent and dusted on the stigma of the female. So once it is over, again rebagging. So why this much things are done? The answer is in order to get accurate result. So he was getting accurate result after doing this much things. That means after performing this one, emasculation, bagging, dusting and rebagging. After performing volume Okay, now after performing emasculation, bagging, dusting and rebagging, you should understand this will be producing the result of exactly the result of these two parents. For example, this flower will be turning into the pod and inside that they will be producing the seed. The seeds will be representing the next generation. When it is put in the soil, they are representing the F1 generation, and what happened is all of them were growing tall. So hope you understood the steps of artificial hybridization and why it was done. Now let's see. Choose the correct option based on the following statement. Emasculation is done in plant which is considered as male parent. No, emasculation is actually done in the plant which is considered as female parent. Emasculation is done in the plant which is considered as female parent. Emasculation prevent undesirable cross-pollination. That is wrong. Emasculation is actually preventing undesirable self-pollination. Emasculation is done to prevent self-pollination. So you can see both 2 and 4 are correct. Hope you got this much. Now shall I proceed to the next question? Can you please comment? Yes. Now next question, match the terms given in column 1 with their example in column 2 and select the correct option. First one, codominance. Codominance means whenever two different alleles are coming and both of them are expressing, that is actually called as codominance. Suppose coat color in cattle you might have learned. A red coat colored cattle and a white coat colored cattle is getting crossed. If both red and white is appearing, that is wrong. Like that, both are appearing in heterozygous condition, that is called codominance. Even AB blood group is an example of codominance. When IA and IB is coming together, when IA and IB is coming together, you can see the surface of the blood cell will be producing both A antigen as well as B antigen. So in heterozygous condition, if both the alleles are expressing or both the genes are expressing, that is actually called as codominance. So here the example that is given for codominance is, the example that is given for codominance is coat color in cattle. So coat color in cattle is an example of codominance. Next one, pleiotropy. If a single gene produce multiple effect, that is actually called as pleiotropy. Example, phenylketonuria. In phenylketonuria, displayed. In phenylketonuria, you can see a single gene will be producing multiple effects. So what is phenylketonuria? Let me tell. In phenylketonuria, you can see it will be having phenylketonuria chromosome number 12. If it is having capital P in both father and the mother, so chromosome number 12, it is having the paternal and maternal chromosome at a locus. If it is having the dominant allele, they can actually produce the enzyme phenylalanine hydroxylase. That is the enzyme that is produced. So name of the enzyme is phenylalanine hydroxylase. This enzyme is produced means surely they will be converting the amino acid phenylalanine into tyrosine. So you may be knowing the structure of phenylalanine. So here it's an aromatic amino acid, phenylalanine. This phenylalanine will be converted into tyrosine by the enzyme phenylalanine hydroxylase. So this is phenylalanine structure, the amino acid structure. This will be getting converted into tyrosine. Tyrosine structure is very simple, same structure only, but it will be having a OH in the aromatic ring structure. So in this one, if you are taking, you can see it will be having one OH here. So if this OH is coming here, so like this, the OH will be coming in case of tyrosine. So tyrosine is actually produced by the enzyme phenylalanine hydroxylase, and this tyrosine will be involved in many activities in our body. Especially they only combine with iodine in order to form thyroxine. So the hormone thyroxine is produced by tyrosine plus iodine, and even the skin pigment melanin formation, it will be requiring tyrosine. Even the hormone adrenaline, the production involved tyrosine. So understand, a person who is having phenylketonuria, these two dominant alleles are absent. In place, the recessive allele, whenever it is coming, in homozygous condition. So whenever the recessive allele is coming in homozygous condition, you can see the problem will be arising. So small p I am writing, that is the defective allele. When this defective allele is coming in homozygous condition, this enzyme will not be produced. If the enzyme is not produced, the tyrosine cannot be produced. Phenylalanine will start accumulating in the body. If phenylalanine is accumulating in the body, brain and all, many problems will be arising like mental retardation. Even so many other multiple effects will be there, like the melanin production won't be there, so there is graying of hair, as well as there will be problems associated with tyrosine. Like that, plenty of problems will be arising due to a single gene. Like this, single gene influencing multiple phenotypic effect is called as pleiotropy. So the second option I will tell, pleiotropy is example phenylketonuria. Now next is multiple allelism. So before telling what is multiple allelism, let me tell what is alleles. Alleles means alternate form of a gene that is located at same locus of homologous chromosome. So if I am taking a homologous chromosome, you know that we have 23 pairs of chromosome. In that, one is paternal chromosome and the other is maternal chromosome. So like that, a pair of chromosomes are coming, one paternal and one maternal. And in that paternal and maternal chromosome, at this locus, what is coming? There is segment of chromosome in that gene only is coming. Suppose in the paternal chromosome in the pair, if stem height gene is coming, so if stem height gene is coming, maternal also at this location it will be stem height. Then at next location, if flower color is coming, here also flower color will be coming in the next one. So like this, the chromosome, the paternal and maternal pairs of chromosome that is carrying almost same characters genes that are actually called as homologous chromosome. So hope you understood the paternal and maternal chromosome. And in the paternal chromosome, at this locus, stem height can have two possibility. Either it can be having a tall allele or it can have a dwarf allele. Here also it can have two possibility. Either it is tall or you can see it is dwarf. Like this, two alternate form of an allele, alternate form of a gene that is coming at same location in the homologous chromosome only, we call it as alleles. Hope you understood what is allele. So normally alleles two only will be coming. If both are looking same, like this, in both locations similar alleles are coming, we call it homozygous. In paternal and maternal dissimilar alleles are coming, we call it as heterozygous. Hope you understood homozygous and heterozygous. Suppose father tall allele, mother tall allele, it is homozygous. Father tall allele from mother dwarf allele, then it is heterozygous. This is how we call it. But just imagine if a character is having, for example, at this locus, just imagine blood group the gene is coming. Blood group the gene is coming. You know that in blood group there are three alleles that are coming. That is IA, IB and IO. Like that, three alleles are coming. So suppose here it is IA that is going to come, or IB, or it will be IO. So like this, more than two form of an allele is coming for a character, that is called as multiple allelism. So this location also, it can be IA, IB or IO. So from this three, any one only will be coming at this location. Suppose it is IA from father, IA from mother, it is a group. So hope you can understand what is meant by multiple allelism. So multiple allelism, you should understand, at a locus if more than two form of a gene is coming, that is called multiple allelism. Human blood group is an example of multiple allelism. So allele means don't simply write it is alternate form of a gene. It is actually alternate form of a gene. Multiple allelism, when it is coming, you should understand more than two forms are coming. That is actually called multiple allelism. So example for multiple allelism that is given here. is human blood group. In human blood group, it is having more than 2 alleles. Now, polygenic inheritance. So whatever I was discussing here, only one type of gene is influencing a character. Suppose a character is influenced by more than one type of gene. For example, human skin color, if I take, in the paternal or maternal chromosome, not only one gene, A, B, C, like that, different genes at different locus in a chromosome all together will be acting for one character. They all actually act together in order to bring one single character. That is actually called as polygenic inheritance. More than one set of gene that is trying to influence or to make a character, that is called polygenic inheritance. So human skin color is an example of polygenic inheritance. So further I am not discussing that. Here the option for polygenic inheritance is 4. So answer 3, 1, 2, 4. Hope you got it. Now next one, a question from pedigree, one application question. Now shaded symbol represent black guinea pig while open symbol represent white guinea pigs. Black skin is dominant over white. So black is dominant. So let's take 2 genotype for black. If I am taking black, it can have 2 genotype, either homozygous or heterozygous. Then white guinea pigs, if you are taking, it's having only one genotype, that is small b, small b. Now let's write and see which one is applicable. Anyway, one will not be, it can be applicable for both male and female. It's not said it's sex linked or anything. So let's directly write down what is possible. So here 1 I can write only small b, then 2 it is, if I write capital B, all their progenies should be affected. Since all the progenies are not affected, we can write it as heterozygous, capital B, small b. Then coming to 1 in the 2, here you can see it can be either, only possibility is small b, 1. Then 2 can be capital B, small b. Capital B from father and small b from mother. And 3 if I am taking, that is also capital B, small b. Then 4 can be small b only. Then 3, 1 can be from the mother, they should get capital B because the disease is there, from the father small b. Then here it can be small b only. So I have written all the genotypes that is possible. Hope you got it. Now let's see the answer. What is 2, 1? This one, and 3, 1, this one. So the answer 2, 1 can be small b, 3, 1 can be capital B, small b. So the answer is 3. Hope you got it. Now next is genes which are tightly linked on chromosome shows. So if you are taking a chromosome, if the genes are located very close to each other, understand they show very low recombination. For example, in Drosophila, page number 84 in NCERT, you might have gone through the Drosophila's experiment conducted by Thomas Hunt Morgan. He was actually learning about the body color and eye color in Drosophila as well as the wing shape. So body color, he was actually learning about the yellow body color as well as white body color and the eye color. So like that, when he was learning about the body color, eye color, etc., you can see certain characters, they are actually located or they show very less recombination. Very less recombination is shown by 2 characters, means understand they are located very close to each other. So if 2 genes are very close to each other, A and B, they will show very less recombination. So new combinations you won't be getting when two genes are located close to each other. Such type of genes which show very less recombination, they are actually called as tightly linked genes. For example, A and B. If the percentage of recombination between them is 6, I will tell they are very close to each other. Such type of genes are actually called tightly linked genes. Such type of genes are called as tightly linked genes. But if two genes are showing very high rate of recombination like 40%, understand they are located very far apart from each other. That is why always they are getting segregated, resulting in new combination. Such type of genes are actually called as loosely linked genes. So tightly linked genes, understand they are located very close to each other, and loosely linked genes, understand they are located very far from each other. So what was said by Thomas Hunt Morgan, if two genes are located very close to each other, they will show very less recombination. So percentage of recombination is directly proportional to distance between the genes, and percentage of recombination is inversely proportional to the strength of linkage. More the recombination, less is the strength. Less the recombination, more is the strength. This ideology has been used by another scientist for understanding the location of genes inside the chromosome. That is gene mapping, which was done by Alfred Stewart Event. So Thomas Hunt Morgan only conducted all these research and all on Drosophila melanogaster. Later, this gene mapping was done by Alfred Stewart Event using this ideology. So here, genes which are tightly linked on chromosome show very less recombination. High recombination is wrong. High recombination means, understand, they are loosely linked. They are located very apart. Very low parental combination is wrong. Tightly linked means very less recombination and more is the parental combination. So the answer is 1. Next one, which of the following statement is correct about polygenic inheritance? In polygenic inheritance, one gene regulates the expression of many characters. You know that in human skin color, 6 genes are coming. If all the dominant genes are coming together, full expression of the trait like this. If 6 dominant genes are coming, the person's skin color will be very dark. Whereas if only if only 5 dominant genes are coming, the color will start changing. It will become fairly dark, like that it will keep on changing. So human skin color is actually a polygenic inheritance. You can see one gene will be regulating the expression of many characters. So 6 dominant genes are coming, full expression of the character, very dark skin color. Only 5 dominant alleles are coming, you can see it will become dark. 4 dominant allele, fairly dark. 3 dominant allele, mulatto. Like that, the pigmentation will start decreasing with the absence of number of dominant allele. So polygenic inheritance for skin color, you should understand it's a qualitative inheritance also. Sorry, quantitative inheritance also. Not qualitative, quantitative. The quantity of dominant alleles will be deciding the character. Then qualitative characters are regulated by polygenes. Then besides the involvement of multiple genes, so it's not inheritance of many character. Here you should understand it will be influencing only one character. Then qualitative characters which are regulated by polygenes. Qualitative, no, it is quantitative. Then besides the involvement multiple genes, polygenic inheritance also takes into account the influence of environment. So understand human skin color also it is influenced by the environment. So this is correct. Then human skin color is an example of not qualitative character, it is quantitative. Hope you got this much. So polygenic inheritance, one gene regulates the expression of many character is wrong. Actually it is many genes regulate the expression of one character. That is the correct one. Then second one, qualitative, it is wrong, it is quantitative. Hope you got this much. Next one, are you getting it? Can you please comment? Okay. Now, in dihybrid cross between capital A, capital A, capital B, and small a, small b, the ratio of, like these questions are coming, it is easy to do. That is, in this condition, what is the F1 generation looking like? Let me show. The gametes they will be forming is this one and this one. And F1 generation, you can see it will be having, F1 generation will be having capital A, capital B, small b. Then when it is selfed, you will be getting a ratio. So I am going to write the genotypic ratio. You might have learned the genotypic ratio from the dihybrid experiment. You may be knowing it is 1:2, 2:1:2, 2:4:2, 2:1, 1:2:1. I'm going to write it. The first one will be capital A, capital A, capital B, capital B. All the one that I am going to write will be double homozygous. Then diagonally in your box you will be getting double heterozygous also, capital A, capital B, small b. That will be double heterozygous. Remaining all, when you are checking in the box, you can see it can be one homozygous and one heterozygous. That will be capital A, capital B, small b. That will be coming 2 times. Then small a, capital B, small b. Then next is capital A, small b, capital B, small b. Like this it will be coming. Then here it can be capital A, capital B, capital B. Then here it can be small a, capital A, small a, like this. So you should understand, whatever I have written in yellow color, they will be coming only 1 out of 16. So how many times they are coming? They will be coming only 1 out of the 16. In the 16 boxes when you are writing, these all will be appearing only once. But whatever I have written in green color, they are all appearing, one homozygous and one heterozygous. They are appearing around 2 out of 16. And this double heterozygous, they will be appearing 4 out of 16. So in a dihybrid experiment, if any genotype is given, easily you can calculate. You don't have to write a lot, and you don't have to find the genotype. You can directly answer. So here capital A, first one, capital A, capital B is given. What you should write? Double homozygous. The answer is 1. Then here 1 homo, 1 hetero. The answer is 2. 1 homo, 1 hetero. The answer is 2. Double homozygous. The answer is 1. So no need to write the entire box and waste the time. You can directly take the answer 1:2:2:1. So this is how you can easily do it. Now next one, match the column 1 with column 2. Alleles. So what are alleles? Alternating form of a gene is actually called alleles. Already we discussed it. Genotype. Genetic composition of a character is called genotype. So that is actually genotype, an identical individual. So if you are taking this genotype, you can see it will be having the genetic constitution of an organism. Genetic composition of an organism is actually called genotype. So alleles are alternate form of a gene. Genotype is the genetic composition of an organism. For example, for tallness, you can write either homozygous or you can write heterozygous. That is the genetic composition. Then homozygous. If two similar looking alleles are coming for a character or identical alleles are coming for a character, that is actually called as homozygous. Then heterozygous. If two different alleles are coming for a character, that is called heterozygous. 4, 3, 1, 2. The answer is 2. Hope you got this much. Shall I proceed? Can you make a comment? 3 and 1 now. Okay, got it. Yes. Now next one. Here, look at here. You can see homozygous condition. It is having 50 centimeter. So genetic composition. But in heterozygous condition, it's not the full characters that is coming. So if a character is coming in homozygous condition and heterozygous condition differently, understand it is a monogenic quantitative inheritance. Best example given in NCERT is the flower color in incomplete dominance. Incomplete dominance, if you are taking flower color in Mirabilis jalapa, you can see red is coming in this condition, but pink is coming in heterozygous condition and white is coming in homozygous condition. So understand, like this and all a character is coming. That means when both the dominant alleles are coming full expression, that means a lot of pigmentation is there. One dominant allele is absent means pigmentation is less and the flower color was pink. So such type of inheritance is monogenic. One gene is controlling, but it is quantitative. So here I should tell A is an example of such type of inheritance which is monogenic and quantitative. But this side you just check B. You can see whether it is homozygous condition or heterozygous condition. Capital T is forming 50 centimeter. Capital T small t is also forming 50 centimeter. So that is qualitative inheritance. It's not quantitative. Quantity of alleles are not deciding the character. One allele is there, full expression of the trait. So the answer is 2. Hope you got it. Next one. Thomas Hunt Morgan experimented on. The answer is fruit fly. So Morgan's experiment was on Drosophila melanogaster, that is fruit fly. On that only the experiments were conducted by Morgan. So almost same reason as that of why Gregor Mendel was selecting pea plants. It was also having a short life cycle. A single mating can produce a large number of progeny in fruit fly. Not only that, it was actually growing on simple synthetic media. In ripened banana and all, we can easily multiply them in the laboratory, and the male and female was easily distinguishable. So these are the reason why Morgan was actually experimenting on fruit fly. Hope you got it. Can you please comment? Yeah, sexual dimorphism was there. The male and female was easily distinguishable. and it was visible under a low power microscope also. Then single mating produce large number of progeny, like that, so many characters. Yes, okay. Now next one, read the following statement. It produce disorder in females more than males. Then all female offsprings will exhibit disorder if father possesses it. So if the father is possessing it, that means it is X-linked. If father's chromosome is XY and mother is XS. For example, if the father is having the defective gene, let me put it as capital D. Always a female child will get one X chromosome from the mother and one X chromosome from the father. Yes or no? So suppose they are getting one from father and always getting the disease, means understand this is a sex-linked dominant gene. Let us check the remaining option also. They are not transmitted to the son if mother does not exhibit, then it is confirmed. It's not transmitted to the son if the mother does not exhibit disorder. Which of the following gene will have? Because if the son is not getting it and the mother is not having it, son will surely get X chromosome from mother only. So if the mother is normal, the son is normal, like that condition is coming. Understand, this is X-linked gene because X chromosome is always going to the son from mother only, and the son will always get only Y from the father. So confirm, the answer is sex-linked dominant gene. Next question, which of the following is not true for F2 generation of a dihybrid cross? Dihybrid experiment you already know, it's not having 9 phenotype, it's having only 4 phenotype and 9 genotype. For example, stem height and the seed shape. If a cross is conducted, they were getting a ratio of 9:3:3:1. In that, 9 will be tall, round, 1 will be dwarf, wrinkled. That is which character? That is parental combination. Whereas you can see remaining will be recombination. That is the tall that is coming with the wrinkled and the dwarf that is coming with the round. Like that you can see new combinations also will be coming. That's all. Now let's see the other options also, like 9 genotypic categories and 16 zygotic combination. That is correct. So first option is actually wrong, that is incorrect. Then 62% is parental combination. 62% means what? You can see the around 10 out of 16 or 5 out of 8 is parental combination, and remaining 6 out of 16 or 3 out of 8 will be which combination? So here you can see around 6 out of 16 or the 3 out of 8 will be recombination, that is 37.5%. Then 4 types of male gametes and 4 types of female gametes only will be obtained from the F1 generation in a dihybrid cross. So if you got this much, we will move for the next question. So already we have discussed this. Genes regulating many phenotypic characters are called. A single gene that can influence multiple phenotypic effect, that is actually called as pleiotropy. Shall I proceed? Yes, Reem. Now next one, substitution of amino acids in the globin protein results due to. So next topic is about the sickle cell anemia. Sickle cell anemia is an example of point mutation, where you should understand a person, if it is having HBA gene, in the HBA gene the DNA will be having the CTC, the 6th codon will be CTC, this CTC will be resulting in the formation of mRNA, that is again GAG. So understand, in the DNA, if there is CTC, they will be resulting in the formation of an mRNA where the sixth code will be GAG. So like this, first code will be coming, second, third, fourth, fifth, and the sixth position it will be GAG. Like that it will be coming. This is the normal case that will be coding for valid. Now you can see whenever there is mutation, this AT will be turning into, TA. If like that it is happening, GAG will be coding for GUG. Like that it is coming when the amino acids are formed. You can see at the sixth position, the wrong amino acids will be coming in the beta chain of hemoglobin. That will lead to the formation of sickle cell anemia or sickle shaped RBC. Even sickle cell anemia, you can see it is due to the defective gene HBS that is occurring due to a point mutation. Yeah, very good, Reem. Yeah, so Venkata has also answered. So here you can see there is a substitution. So here you should understand AT is becoming TA. That is a point mutation. In that it is a substitution, and in that it is a transversion. Hope you got it. So the answer is CTC is turning into CAC. Flower color in Mirabilis jalapa is due to, already we discussed, it's a example of monogenic inheritance as well as quantitative inheritance, where you can see when two different alleles are coming in heterozygous condition, one will be partially expressing and the other is suppressing. So you can see an intermediate character is formed. So 4 o'clock plant, Mirabilis jalapa, snapdragon, etc. you can see it's an example, or flower color is an example of incomplete dominance. Hope you got it. Shall I proceed? Yes, snapdragon. Next one, distance between gene on a chromosome representing 12% crossover. So understand, in a chromosome, if there is distance between the two genes are 12%, understand they are having 12 map unit, that is the distance, because according to Thomas Hunt Morgan, percentage of recombination is directly proportional to distance between the gene. So if two genes show this much percentage of new combination, understand that is the distance between them. This ideology was used by Alfred Stewart Evans. Next one, in a typical monohybrid cross, we have crossed a hybrid tall plant with another hybrid tall plant. Which of the following result cannot be expected? So one tall plant, if crossed with another hybrid tall plant, capital T small t, the result will be what? 3:1. 3 tall is to 1 dwarf. This will be the result. And in this 3, you can see 1 will be homozygous, 2 will be heterozygous, and this one will be homozygous. 1:2, 2:1. So 25% are homozygous tall. That is correct. 25% are homozygous tall. and 25% are homozygous dwarf. That is also correct. 25% are homozygous dwarf. Then 100% is heterozygous tall. That is wrong. Then 75% is tall. So 3 out of 4 is tall and 1 out of 4 is dwarf. So this is also correct. So only 3 is incorrect. So that's all about today's question discussion. Hope you understood the concepts and all. So let me wind up the session, and in advance let me wish Happy National Sports Day of Qatar. Okay then, bye.